let's start off really easy, right? So super, super basic anatomy. So when we're looking at our basic anatomies, our heavy lifters, what are these guys? Kidneys, right? So kidneys connect to what? Two tubes, right? What are they called? Ureter. Don't call it a urethra, right? Different things. So ureters go to the bladder, which then go to the urethra, right? So very, very basic anatomy when we're working with our organs. So, 3 functions of the urinary system. There are actually quite many more functions of the urinary system, but 3. What is the heavy hitter for urinary system? What's the primary function? Making urine, right? Urine production, obviously. What are some other ones that we could put in? Blood pressure, right? Blood pressure regulation, right? We saw that Wednesday. What else could we do? What else can we regulate in the blood? pH levels, right? So pH levels, making sure our blood is not too acidic or too basic. We have our blood pressure regulation. We can even say some more broader concepts like ion homeostasis, right? Because we saw that we definitely are going to be shuttling a lot of ions in and out of the urinary system, as well as maybe like endocrine hormonal production, right? We know that the kidneys are spots for things like calcitonin, or calcitriol, as well as things like erythropoietin, right? So managing our levels of erythrocytes by erythropoietin. So there's actually quite a different variety of functions for our urinary system. Our innermost connective tissue layer. So what connective tissue layer directly touches the organ? What do we think? So when we look at the kidneys, right, it looks like it's coated in a very thin layer of something. What do we call that? It's a opposite of that one. It's a renal, starts with a C. There's a capsule. Corpuscle is inside, right? So we have a renal capsule, right? So when you guys physically go into lab, whenever we're done with our GI system, basically the dark red is called the renal capsule. So it's a thin layer. Do we remember the actual type of connective tissue? It's all going to be the same other than your middle layer, which is adipose. So dense, irregular, right? So this should make sense because dense, irregular, right, usually is really tough. So we have a very thin layer of dense, irregular connective tissue that's coating that kidney. And if we flip it up, so the outermost connective tissue layer, now you can say it. What's our outermost? That renal fascia, right? So fascia means very, very thick connective tissue. So in this regard, it's very thick, dense, irregular connective tissue. So what might be the differences in their function? So what's our primary function of our renal fascia? What do we think? If I have all of that thick connective tissue, what's its primary job? To hold it in place, right? So we have to make sure the kidneys are situated correctly in the abdominal cavity. And then we kind of just saw this in our little diagram, but how do we connect to the bladder? What are ureters, right? So again, I see it every semester. Sometimes we'll switch the words ureter for urethra. So ureters come first, and then the urethra comes after the bladder. Okay, lovely picture of a sagittal cut. So let's see what we can find here. So you guys can actually see that renal capsule, right? So this really thin, kind of darkened layer is the capsule, that really thin portion of dense irregular connective tissue. So what are we working with when we're looking at our regions? So what is our outer kidney called? Cortex, right? So anything outer, so we see this a lot in organs, we'll call that outer portion the cortex. And then all of this inside, what do we call? Medulla, right? So inside is going to be medulla. Now, let's say, what's the most important thing we can find in the cortex? So all of this stuff, we'd have to look microscopic, but why do we care about the cortex of the kidney? It's where, right, and this is how we filter urine. That's the very beginning portion of filtering our blood. So our renal corpuscles, right, the very beginning, the glomerulus and the Bowman's capsule, are all going to be in the cortex. Then we can, if we remember, there are some nephrons, that usually are extensively in the cortex versus the medulla. So we definitely care about the cortex. Now when we go inside the medulla, what do we call these giant kind of triangle wedges? Pyramids, right? So we have our renal pyramids. Now, class yesterday did not know. So does anyone remember what do we call, in anatomy we call it a bump. So what is our word that we've learned that literally means a bump in anatomy? Papilla, right? So like the bumps on your tongue, right? So our renal papilla are going to be the entrances into our calyxes. So kind of the exit region of the pyramid, entrance into our calyxes. Now, when I'm looking at my calyxes, do I start with minor or major first? These guys are smaller, right? I can physically see these little areas are smaller than something like this. So these got to be minor. So these are minor calyxes. They're then going to drain into our major calyxes. From there, where are we draining? So what do we call this little area? You're very close. The hilum is this indentation where we grab all three of these kind of vessels. So you are close, but it's right before we get to the hilum. So this is called our renal pelvis. So we drain out the renal pelvis, and from there, we're then going to go into the ureter, right? Now that leads us to the hilum, right? So this is what we like to say the entrance and exit point of the kidney, right? I see my two prominent artery and veins, so renals, right? And then again, that ureter. So let's see if we've covered everything. That looks like our major structures for macroscopic structures. So we're comfortable with, right, what we can find inside the cortex, what we can find inside the medulla, pathway of urine from a macroscopic standpoint. Okay, so this is also a really good one. So switching up, we also saw that we had to know the vasculature because, right, we have to know how to get to a renal corpuscle. So if our goal is to get into a renal corpuscle, right, we just saw the very beginning steps. So of course we're going to have paired arteries and veins. The veins are going to be going away, so let's just go with our arteries. Our renal artery, as we go through and we go into the kidney, we see that it's eventually going to branch off. So what is our first branching point inside the kidney? What do we call that? It starts with an S. So we branch into segments, right? So these guys are segmental arteries. So we have segmental arteries, and then we can see, right, if we follow these segmental arteries, they're going to eventually go between the lobes, i.e. Between the pyramids, our renal pyramids. So what will we call this? Interlobar, right? It literally tells us in the name, inter for between, lobe for lobe of pyramid. So from here we see interlobar arteries and veins. Now as we travel, let's just pick this little area. So as we go through interlobar, I see that eventually I'm going to start hugging, right, the cap of that pyramid. So I make that lovely shape that should remind you guys of the McDonald's shape. So what do we call those? The arch, right? So it sounds just like arch. So arcuate, right? So arcuate means to arch. So we have our arcuate arteries and veins, and then our last destination before we'd have to go microscopic, all of these little guys, right? So it looks like they radiate out from the pyramid. So we call those cortical radiate. Cortical for cortex, radiate for obviously it looks like that they're coming out and they're making rays. You can also call them interlobular. So again, you guys will hear that a little bit more in lab. Just don't confuse it with interlobar. Now, critical thinking, right? So as we go through our cortical radiate, even though we can't see it, what is the next step? Once I go through cortical radiate, where am I going? The what? Yeah, the afferent arterial, right? So at that point, we have to switch to a microscopic view because these guys will lead into the afferent arterioles, that leads into the glomerulus. We understand, right, the flow from there. Okay, beautiful picture of our renal cortex, right? So that outside region. Now, when we're looking at our renal cortex, there are two prominent structures that we have to care about, right? We have all of these big circles, right, that look like they've got all this stuff going on inside of them. And then we have all of the background. So if we focus on these guys, what is the proper name for one of these structures? And it might be a little bit confusing. So sometimes we want to say glomerulus, but that would not be correct. It contains the glomerulus, right? So So do we remember the difference between a glomerulus and a renal corpuscle, right? So what we're seeing, all of this is considered a renal corpuscle. The difference is a renal corpuscle is the glomerulus, which is what's inside, plus the Bowman's capsule, which is the little encapsulating thing that has the Bowman's space. So altogether, we call this structure the renal corpuscle, right? So I can see at least 3 good ones, and there's one over there as well. So I have 3 to 4 renal corpuscles. I see the glomerulus. I see my Bowman's capsule with my Bowman's space. Now we mentioned what epithelia we are expecting, and we can even see it. Usually it's the best to see it when you look at the Bowman's capsule. What is this epithelia? Very important. Simple squamous, right? We have to understand why. What is the job of simple squamous? It's filtration or diffusion. At this moment in time, it is filtration. So we're filtering blood, that makes sense. Now, all of the background, right, I should also care about. I know it's a little bit fuzzy on the picture, but we can see all of these backgrounds they look like little cuts right through circles, all these little circles. So what are we looking at? What is all these little kind of half cuts? What do we think? What else is in the kidney other than the renal corpuscle? What is everything else of our pathway? We've seen it extensively, all the tubes, right? Proximal convoluted tubule, distal convoluted tubule, that's the majority of our cortex. You guys should know this, right? So all of these are the tubes that urine or your filtrate is going to be eventually flowing through. They're just using cross-sectional cuts so we can see kind of what's on the inside. You can see the filtrate or the tubular fluid flowing through them. So because this picture is hazy, I wouldn't really want you guys to know the difference between proximal versus distal convoluted tubule, but you should know that the major difference between them is one of them has an additional structure that helps out. What does the proximal convoluted tubule have that our distal convoluted tubule does not? What is it? So microvilli, right? So our proximal convoluted tubule has all of that really good microvilli so that we can come in contact and reabsorb things and secrete things. So proximal convoluted tubule usually is more what we call kind of hazy, but from this picture, they all kind of blown up. So we're not going to worry too much about identifying the differences histologically there. So we can also see the epithelia of each of the tube, right? We know what the epithelia is. What's the epithelia of all of our tubes? Cuboidal, right? Simple cuboidal. Now, if we do remember, right, there is a portion of the loop of Henle that turns into simple squamous, but all of your tubes, your proximal and your distal, are going to be simple cuboidal because they function in absorption and secretion. So we have to understand chapter 5 from last semester because it comes back right with a vengeance. Okay, so we're comfortable with our histology for our urinary system. So, why are our ureters capable of peristalsis? So if the ureters just connect to the bladder, right, and we use gravity to push urine down, why do I also need to have the capability of peristalsis? So what happens when you're no longer standing up straight, right? So when you're sleeping, right, you guys remember that example, when we're sleeping for 8 hours, we are no longer in that vertical position, right? We're now horizontal. So if our ureters did not have the capability to go through peristalsis, your urine would just get backed up, right? It wouldn't be able to leave the kidney. So definitely we have to have a way of physically pushing things forward so that we can get into the bladder even when we're not in a vertical position. Our ureters and bladder are what sort of epithelium? What do we think? So we saw this last semester, right? We also saw this in this chapter. So what's our bladder? Transitional, right? And the ureter is the same thing. So both of these guys have to accommodate, right, for the filling up of urine. So we have what we call contracted bladders, slides in histology, and distended bladders. We remember, right, those kind of plump cells, they're very plump when when there's no urine, and then they kind of flatten out when we get more and more and more input inside. What anchors our ureter to the abdominal wall? Oh. So you guys remember, we're doing, right, GI extensively in lab, so this should still be on your minds. You guys remember in the GI system, there's 4 layers, correct? Right, you've got your mucosa, et cetera, et cetera. We saw in the urinary system, we typically have 3 layers. So which of the layers is going to anchor? Another way of saying that is, right, if it's anchoring, what's just the outside layer? What do you think? So what is the last layer? Adventitia, right, serosa, adventitia, that is the connective tissue layer. Definitely can't be mucosa, that's the innermost layer, that's epithelium. Can't be, this guy doesn't have submucosa. It can't be the muscle layer, right, that's not the job of a muscle. So it has to be the connective tissue layer, or serosa adventitia. And as we go down, so once we get to our urethra, so at the end of the urethra, what epithelium can we expect to have? Stratsquamous, right, definitely stratsquamous, because if it is an opening, I have to have protection, right? I'm going towards the outside environment, therefore I need to have protection. What's our difference between male and female urethra? So we can stop there. So what is the primary difference between male and female urethras? Length, right, so we're looking at, again, like 3 versus 20 centimeters. And then our implications of this, what about, why do we care? So, okay, let's do, so for female urethra, what is it a lot easier to do? To pass the, so that's the opposite side, right, to pass things, right, so pass kidney stones, pass anything like that, and get infections, right, because it is so short. It's a very short way for a bacteria to just come on up there. We can also say, which, is it a male or female, which urethra is just for urinary system? Female, right, so for male, right, it's both urinary as well as reproductive. Okay, so good picture of a nephron, right, so microscopically. So let's do our vasculature first. So how are we getting into that glomerulus? We know this. So what leads inside the renal corpuscle? This little guy. Afferent arterial, right? Afferent to go inside, to go towards. I'm going to bring in the blood via my afferent arterial, right? We are going to filter it out at the glomerulus, and I'm leaving via that efferent arterial. So we also see as we go down, right, we go into this extensive capillary network that coats the rest of the neuron. What's its proper name? Starts with a V. We know it's so vasa recta, right? So our vasa recta coats everything. So we understand that because we have to have reabsorption and secretion through this entire pathway. So we have to have blood capillaries that are also here. So, switching up to our tube side, I know that again, this is my renal corpuscle, that's the glomerulus, this is that Bowman's capsule. We're filtering blood, so at this point in time, the product is called filtrate, right? Now, as we go down our regions, right, what is this first region we get to? What do we call that? PCT, right? Proximal convoluted tubule. So proximal means closer to the origin point, and we think that this is basically the origin point. So proximal convoluted tubule. Then I go down, right? We see the lovely loop of Henle, right, or nephron loop. We've got that descending limb and then our ascending limb. We go up, we see the distal convoluted tubule, and then we're going to drain into this tiny little area called that collecting tube. All of these are collecting tubes, which then go and drain down our collecting duct. So we understand, right, our different pathways of getting our fluid through. At this point in time, so when we're going through PCT, DCT, and our nephron loop, what do we call that fluid? Do we call it urine? No. Technically, do we call it filtrate? Not really. Filtrate is at the very beginning. So what do we call all of this that travels through? Don't think too hard, right? It's just tubular fluid. It is fluid that goes through all of these tubes. When do we start calling it urine? What do we think? Right, so after it goes, right, drains down the collecting duct, and once we get into those like calyxes. So after collecting duct, we're now going to call it proper urine, because even now at this, when we're going through through that collecting duct, we are still doing things like reabsorbing water. So once it exits that, then we can call it urine. Okay, so good picture. So do we remember, what is this picture showing us? What does it look like it's showing us a difference in? So our different types, right? Different types of our nephrons, right? We saw that there are two different types of our nephrons. We have one that has most of its body in the cortex. So do we remember what we call that one? Cortical, right? So literally just named after the cortex. This is a cortical nephron. And then we have this guy that extends way, way down, right? So what do we call this one? Juxtamedullary, right? Medullary meaning from the doula. So juxtamedullary nephron. Which one's job is to concentrate urine? Which one do we think? Juxtamedullary, right? You guys should understand why that makes sense. So I understand that this one's job is to concentrate urine because it has such a long loop of Henle, right? Even in this picture, I see the change in the epithelia, right? I see so much simple squamous epithelia. Why does it make sense? You guys need to think about why that makes sense in regards to concentrating urine. If I have so much simple squamous epithelia, that means water can diffuse right out, right? So if I take water away from the equation, that will concentrate the solutes that's inside of there. So really good for concentrating urine when we have our juxtamedullary nephrons. Do we know our ratio about how much of our nephrons are juxtamedullary? Is it the majority or minority? Minority, right? We're working with about 15%. Okay, so in the collecting ducts, right? So as we go through almost to where the exit point is, we have two special cells. First, our principal cells. What do they respond to? We can use kind of a broad overview when we answer this. Hormones, right? So that's a perfect answer. Principal cells respond to hormones. They respond to hormones like ADH and aldosterone, right? So they are the cells responsible for having receptors for things like inserting more aquaporins or inserting more sodium pumps to regulate our water reabsorption. And then what about those intercalated cells, right? So remember, we're not in the heart. So what do our intercalated cells do? Instead of working with hormones, what are we regulating? pH, right? So intercalated cells are all about blood pH. And we remember there are two types, right? So type A and type B. So you guys will have to remember which one they do because they're basically opposite. So type B intercalated cells, what way do you think they're going to go for blood pH? Are they going to make the blood more acidic or more basic? So I'll answer it for you. If type B cells put hydrogen in the blood, are we getting more acidic or basic? Acidic, right? So if I am physically putting hydrogen ions back into the blood, we are going to be more acidic. And then we have our other guy, our juxtaglomerular apparatus. It's made up of two different regions, right, or two different cell types. We have cells that are a part of the afferent arteriole. We also have cells that are a part of the distal convoluted tubule. So do we remember the names for them? Any of those names? We have macula densa, right? That's the portion of the distal convoluted tubule. What about the afferent arteriole? What do we call them? Granular, right? You can even get easier than that. We can literally just call them JG cells, which stands for juxtaglomerular cells, right? And then in the grand scheme of things, why do we care about the juxtaglomerular apparatus? What do they work in doing? They're going to regulate are they regulating pH? Are they regulating urine? No. So what's our third option that we harped on? If it's not pH level, it's not making urine, what's the third most important function of the kidney? Blood pressure, right. Now we can even go so far as to say this, you guys should be able to put two and two together now. Specifically, this area is responsible for making and secreting renin, which we saw last class. So we all went through that renin-angiotensin-aldosterone system, right? Now we can see what area of the kidney is responsible for making it. So guys make renin. They are responsible for raising your blood pressure. What is hypervolemia? So break the word down. Hyper means what? Too much, right? So emia means in the blood. So anytime you see emia, that means blood related. VOL, what do we think that means? Again, don't think too hard. What does it look like it's trying to spell out? Volume, right? So meaning too much fluid in the blood or excessive blood volume. Usually we kind of colloquially say too much water in the blood, right? Why could this lead to renal failure? So if you have too much water in your blood, why do you think that can kill your kidneys? What's it going to force on your kidneys, right? What are your kidneys going to try to do? So your whole job of your kidneys, right, is to filter blood. If we have way too much fluid in our blood, they're going to try to get that fluid out, right? So they're just going to excrete it into your urine, correct? However, when we have hypervolemia, when it's just, again, excessive amounts of volume, excessive amounts of water, that means that your kidneys are working overtime, right? They are working and overworking themselves to filter out everything and to put it into the urine. So whenever, it's not just pertaining to the renal system, whenever you have an organ that is overworked, it leads to failure. So liver failure, right? When your liver is overly trying to break down toxic chemicals and alcohol, that leads to liver failure. Same thing for your renal system. So whenever you basically are overworking that tissue, it can overwork itself to destruction. Okay, so 3 steps of urine formation. So urine formation, right? We saw there's 3 major processes. I'll say the first one. So first, obviously, we have to go through filtration, right? We got to filter out the blood. What else do we have to do after filtration? Reabsorption, right? So we have to reabsorb. We have to take back the things we want to keep, right? Put it back into the blood. And then what else are we doing? Secretion, right? So we put things into the tube system so that they can be excreted into the urine. Now, when we're talking about our filtration barrier, so that glomerulus, right, the Bowman's capsule, all that good stuff. We have 3 different layers, right, that allow for really, really selective permeability, so high selectivity. Our first one, right, is just the nature of the endothelium, right? Endothelium, because it's fenestrated capillaries, it's not letting absolutely everything in, but it's going to let in large molecules, but it keeps the cells out, which is the good part. What do we go through after we hit our endothelium? So we have, right, the basement membrane. So after endothelium, I hit that basement membrane. So that's going to be even more selective, usually only smaller things like smaller proteins. And then, do we remember the name of the cells that coat the glomerulus? They look like little amoebas. They're named after their feet. Podocyte, right? So pod for foot. So our podocytes, right, they lock into place with one another, so they create that 3rd additional layer for our filtration barrier. So if we were to have a net filtration pressure of 0, what does that mean? It's equal, so this is telling us something extremely important. If I have a net filtration pressure of 0, am I making urine? No. Net filtration pressure, do you guys remember when we looked at there's 3 pressures, right, inside the glomerulus? You have the driving pressure, and then you have 2 opposing pressures, right? If they equal 0, so if you don't have a positive value, that means nothing can exit the glomerulus, right? If nothing exits the glomerulus, I'm not making any urine, right? Nothing is being filtered. So if I have a net filtration pressure of 0, I'm not making anything. And a part of those 3 pressures, so we saw, right, 3 pressures. We have, let's just go through them. We have the pressure, the hydrostatic pressure of the glomerulus. That basically means, right, that's our driving pressure. That's the pressure that is trying to force things out of the glomerulus. I also have the pressure of the Bowman's capsule, right? So that's an opposing pressure, so the pressure that's trying to push things into the glomerulus. And then I have this 3rd one. Usually this one gives students the hardest time. Blood colloid osmotic pressure. This is a 2nd pressure that is in the glomerulus. So the glomerulus has 2 pressures. one of them drives, the other one opposes. This is the notion that it is the pressure exerted whenever you see this in relation to blood colloid. Basically, it means pressure from everything solid in the blood. So when we think about this, right? In the blood, there are cells. In the blood, there are proteins. There are molecules. All of those things are solids. Because they are solids, you can think of them as a solute, correct? Right? Basic chemistry. So, is this going to be a driving pressure or an opposing pressure? What do we think? It's gonna oppose. You guys have to know why, right? Even though it's a pressure inside the glomerulus, it fights against the driving pressure because all of this stuff is solid in the blood. I have all of these cells, I have all of these proteins, right, that are being kept in the blood because they can't be filtered out, right? So they're all there. What does that mean in regards to water? So in that Bowman's capsule, right, everywhere in your body, you're gonna have fluid, you're gonna have water. What does that mean in relation to the water inside of the Bowman's capsule? Where does it want to go? It wants to go into the blood because we mentioned water always wants to dilute, right? So in this area, if I have a high amount of solid things, water wants to come inside so that it can dilute. If I have water from the Bowman's capsule wanting to go inside the glomerulus, you guys can see how that's an opposing pressure, right? So make sure you understand those three pressures and if they're driving or if they're opposing. So two of them are based off of the pressure exerted by water. One of them is the pressure exerted by solid matter. Okay, so vasoconstriction of our efferent arteriole will do what to our glomerular filtration rate? So our rate of filtration, if I constrict my efferent arteriole, do we think that's going to raise it or lower it? It's going to do what? Raise it, absolutely, right? So why is it going to raise it? What do we think? It's kind of twofold, right? So if I constrict the blood vessel that exits the glomerulus. So there's twofold. One of them is that means that it's harder for blood to leave, therefore blood spends more time in the glomerulus, right? So it spends more time being able to be filtered. And two, if I constrict this guy and it's harder to get out, it raises the pressure inside of that glomerulus. So if I have a higher pressure inside the glomerulus, we just saw, that's going to have a driving force. Most reabsorption in our urinary system, where is that occurring? We mentioned it at the very beginning. One location, right? Is it the loop of Henle? Is it our collecting duct? Where are we looking at it? Has to be PCT, right? PCT, we just said, has the extensive network of microvilli. So most reabsorption, even though you are going to have a little bit in other areas, most of your reabsorption, nearly all of your ions, all of your like vitamins, good stuff, are reabsorbed at the proximal convoluted tubule. Okay, so this picture is showing us some good information, right? We saw that there could be some different factors in our rate of transporting things to be reabsorbed or secreted. We saw two different types of transport, right? If I have transport that goes between cells, what do I call that? Paracellular, right? So between cells, usually a little bit easier, right? So I just squeeze through, most of the time, smaller things. What about if I can't do that, I have to go through the cell, right? So what do we call this one? Through, across means trans, right? So transcellular transport. So typically this is going to be involved with needing the help of other like channel proteins or integral proteins in order for you to get through both of those membranes. And then another good thing that we can see is the reason why it is so easy to reabsorb water. So we mentioned, right, one of the major goals of the urinary system is to reabsorb nearly almost all of the water that gets put inside. So of course there is water that comes out in our urine, but in the grand scheme of things, there was a lot more put in at that glomerulus, and then we basically reabsorbed the vast majority of it. The reason reason why it is so easy to reabsorb water is because of the pressures inside of the capillary beds of the vasa recta. So, low hydrostatic pressure, that means I don't have a lot of water in these areas because most of the water has been filtered out, correct? So if our water is traveling through the tubes, that means that I don't have a lot of water traveling through the capillaries. So, low water pressure, but high colloid pressure. So if I don't have a lot of water, I have a lot of solid things in the capillary beds. My cells that couldn't go through, all of my big proteins that couldn't go through, right? So as we just mentioned, because we have a lot of solid things and we have very low water in this area, it creates a really high driving force for water to want to diffuse into the vasa recta, right? So that we can dilute things and we can bring back up the water level or the blood volume. Okay, so if I have too much sodium in my blood, what do we call that? So we already saw, so too much, what do we say in anatomy? Hyper. Sodium, Na, right, Na. And then again in the blood, what do we say? So we saw the emia, right? So you just add in basically a T. So hypernatremia, right? So excessive sodium in the blood. Why does that present with CNS problems? Or basically when you have any sort of imbalance in sodium, why do we see CNS problems all the time? Sodium is used in depolarization, right? So if I have an excessive amount of sodium or too little sodium, right, your neurons are not going to be able to work. If I have excessive amounts of sodium, that typically means that your neurons can start excessively firing when they're not supposed to be. Our normal pH of the blood, what are we working with? Right, so almost neutral, 7.4, we're slightly basic. If sodium exits the blood, what does it do to pH? So this is going to rely on your knowledge of understanding that sodium uses a counter-transport channel protein. So if I say sodium uses a counter-transport, that means that whenever sodium goes one direction, something else has to go the opposite side, right? It's a counter-transport. Very important to understand that sodium pairs with hydrogen. So if sodium is leaving the blood, what is it going to do? What is hydrogen going to do? Sodium leaves the blood, hydrogen comes in, right? Because that's the nature of the sodium hydrogen countertransport protein. So what will that do to pH? Raise it or lower it? Lower, right? Remember, to lower is to get more acidic. So if hydrogen is entering the blood, we're getting more acidic. That lowers blood pH. Lower scale is for acidity. Blood that is too basic, what do we call that? Alkalosis, right? So we have acidosis, which would be if we have too much of this happening, and then alkalosis is when we have that basic blood. Now we saw our lovely, right, regulatory system for maintaining blood pressure, right? So very, very important for maintaining blood pressure. Usually when we have a dip in our blood pressure, our kidneys have to release that beautiful enzyme, renin. So renin, we learned from last class, goes through and basically modifies a protein, right? We're going to cut, does anyone remember the first name? It's long, but right, it basically tells us what it is. Angiotensinogen, right? OGEN means it's very inactive. Angiotensinogen, our renin is going to cut it into angiotensin 1. Do I use angiotensin 1? No, I gotta modify it again, right? So that's why we have that ACE enzyme, our angiotensin converting enzyme. That'll cut it again into angiotensin 2. Angiotensin 2 does that entire after effect, so the downstream effects of what we want. So we can just have, right, things like vasoconstriction. So if I constrict my blood vessels, I'll raise my pressure. I can stimulate the release of ADH. I can stimulate the release of aldosterone, right? Why do both of those also raise blood pressure? What are they doing in the grand scheme of things? They're putting more water, right, into your blood. And we mentioned how putting more water into your blood will raise your blood pressure. You're physically forcing, right, water into a finite space. So that will raise the pressure of the vasculature. Now you will have to also remember, right, there is a difference in how they do that, right? ADH, aquaporins, aldosterone, sodium, potassium pumps. Okay, so our last little bit. So how does renal ptosis cause renal failure? So is the movement of the kidney itself the issue? Not really. Your kidney can drop and still work just fine, right? So is it really because it doesn't just drop overnight, right? So it takes a while for you to degrade the adipose and for you to degrade the connective tissue. So the movement over time of the kidney is not the issue. What is the issue? It's the kinking of the ureter, right? So as the kidney drops or droops, it will kink the ureter. When the ureter is no longer in that straight position, right, it will cause a backup of urine. So if we cause a blockage and a backup of urine, that can definitely lead to damage in the kidneys and cause renal failure. What do we call kidney stones? Ooh, renal something, right? Renal calculi, right? So kidney stones. Typically, as we mentioned, if they're really tiny, we can pass them. If they're bigger, we have to do things like lithotripsy, right? Or basically having surgery in order to break them down. Differences in urethral size, primarily, yeah, so what disorder is usually caused by a difference in urethral size? What do you think? UTIs, right? So women extensively get them. It's very uncommon for men to get them because, again, of the size of the urethra, right? What bacteria is usually UTI? E. coli, right? Okay, so that will wrap it up. So make sure you guys do some studying and don't just forget everything.